हेलो टेस्टिंग आ रहा है ऑडियो लेवल चेक टेस्ट 1 2 3 टेस्ट testing 1 2 3 today we'll be talking about single cell RNA-seq lecture 2 of and session 2 of the bioinformatics module of BIO-4505. ये डिले क्यों आ रहा है? हाँ, अच्छा ये टेस्ट था. ठीक है, ठीक. ओके. एंड दिस बैटरी ये चलेगा ना? मतलब 2 घंटे प्री लंच. लंच में एक काम और करना है. लंच में मुझे अपना आईडी कार्ड चाहिए होगा, वो वहां पर है अभी. हाँ, वो ले आना. कोशिश नहीं, ले आना. मैं खाऊंगा कैसे फिर लंच? चल रहा है. एक सेकंड, इससे भी ट्राई कर लो. हाँ, कर दो. हाँ हाँ, कर दो. ये ऑन नहीं हो रहा माइक. ये ऑन यहाँ से नहीं होता. ठीक है. ये कैसे ऑन होगा माइक?
ये माइक, ये छोटा बटन है.
हाँ.
तो लॉन्ग प्रेस करने पे ये ऑन हो गया.
यूजर में.
हाँ. चेक. हेलो हेलो.
तो
यहाँ
टेस्ट
से
कैसे कर?
दिखा रहा होगा.
कैमरा नहीं, माइक का पूछ. मान लो कोई जूम से या क्लास में कोई क्वेश्चन पूछेगा.
क्लास में तो कोई किसी तो माइक पे कनेक्टेड नहीं किया.
ऐसे. ठीक है.
ये है.
करना
अभी आप
टेस्ट.
माइक.
नहीं, उस
हेलो चेक. माइक चेक. हेलो हेलो चेक चेक.
दोनों इसी इनपुट. बेसिकली ये माइक और ये माइक दोनों एक ही इनपुट.
सब एक ही इनपुट पे है, लेकिन बस जो बिना माइक का बात करेंगे ना कोई आवाज नहीं है. आवाज.
ओके. हेलो टेस्टिंग टेस्ट 1 2 3 टेस्ट लेपल माइक. ओके. Testing one, two. Testing one, two. Hello, hello. Testing, testing. And you can hear me right on the mic now. Feel bad because Shireen, you know, and is she not going to any of the other classes also? Zoom. Hello, hello, testing. Hello, testing. Hear me? Morning. You, I know you study cancer and stuff. You are from not a biology background, right? Oh, oh. Have any of you done sequencing in the past or analyzed sequencing data? Beneath you? Did you guys get a chance to watch the video? And fairly easy to understand, right? Not too hard. You you are bio and CS or bio and? You are PhD or I forgot. PhD bio. Have you done any sequencing? Like DNA sequencing data or RNA? Not the. So they will do the wet lab part and they will give you a preparing the library and. But that was before Ashoka, before coming to Ashoka. Here, what lab do you work in? Or no, which professor? Who you are rotating? So you're still fixed. Chemistry. What is this area? Oh, like sleep. Krishna analysis. Corona. Yeah, should we get started or should we wait another 5 minutes?
Professor, I can't hear you.
Yeah, Shireen, we are still waiting for some people to trickle in.
Okay, okay.
Good to have you on Zoom. Yeah.
Yeah. I can hear you now. Yeah.
Okay. Yeah, I'm mic'd up, but yeah, sometimes. your name? Shubham. You are Shubham? You had sent the audit request. You saw my response, right? Effectively, if you are formally auditing, there's no difference between what you need to do. You'll have to do whatever people who are taking for credit have to do, right? Like do all the assignments. And so up to you whether you want to take it for credit. I think it's 10:40, so let's get started. Irene, can you hear us? Hello?
Yes, sir.
Irene?
I can.
Okay, great. Just I'm gonna yeah, feel free to I'll pause at some points. Feel free to ask questions, or
Okay.
if I can't pause, just type them out in the chat, and I'll periodically look at it.
Okay, perfect.
Great. So welcome to lecture 2. I think I've asked you individually, but I'll take a quick straw poll. How many of you have either done wet lab sequencing or analyzed sequencing data in the past, like before this class? You have? What kind? Like DNA, RNA? Huh? NGS, using Illumina. And did you do the wet lab bit or the analysis? Huh? Dry lab. So like you got a BAM file or a and you wrote Python code to do it, or no, what tool or package did you use to BAM? So you are comfortable. Did you do aligning and all that? So for most of you, I think we have a fairly diverse makeup of the class. So we'll assume that most of us have not done it. And feel free to correct me on the wet lab bit, right? I included the second video, which was by the professor at UCSF. I haven't done any wet lab, like preparing a library. I conceptually understand it, but in this class, we'll focus more on the analysis. And assuming you have sequence, you have raw data, how do you analyze, right? It has applications in differential gene expression. You may have a wet lab hypothesis, like this gene is implicated in cancer or some disease, Alzheimer's, right? And perhaps in the second half, I'll show you people have built cell atlases, which is a collection of single cell data across tissues and organs. So often, if you have an hypothesis and you want to take a quick look at it, there are tools out there, these cell atlases, single cell atlases, where you can already, which is like, imagine like a corpus of single cell data for humans and also some other model organisms, which is, you can, it's organized by organ and tissue, and you can take a quick look, like, is, TP53 implicated in cancer, for example. I mean, it can get way more interesting and complex than that. So we'll take a look at that in the second half. Quick show of hands. How many of you, be honest, how many of you watched the 2 required videos? You didn't? Okay. Everybody watched pretty much the Josh Starmer one? Okay. Okay. Just make sure you give me your email after the class. I'm assuming you're not on Google Classroom also. Have you formally enrolled? But you didn't send it to me, right? You send it to Sudip. Okay. Anyway, okay, I'll make sure. But did you watch the video? Okay. So we'll do a quick run through of the video, just so everybody catches up. I think good job. Everybody It's okay if you didn't fully understand it, especially those from a non-biology background, but we'll quickly recap, right? So some of the things we'll not go too much into detail, like how does a sequencer work, but we'll briefly talk about how a sequencer is made of flow cells. So let's get started, right? So I have some slides from Professor Wang at University of Wisconsin. So right, everybody knows the central dogma, right? DNA is converted to RNA, or to be precise, messenger RNA, and messenger RNA is converted into protein. And proteins are kind of, are the workhorse, right? They determine how our tissues work, how our organs work, critical to life, right? And gene expression is critical, right? You know, gene expression is a proxy to measure RNA transcription levels, right? And they can differ across tissues, they can differ across organs, they can differ across normal, healthy state versus pathological or disease, right? And gene regulation is which and how genes are expressed, right? So single cell RNA-seq, the reason a lot of the class focuses on that is that it is critical to understand how genes are differentially expressed between normal and disease tissues and organs. Right? Why study single cell, right? So within a particular kind of tissue or organ, right? For example, skin cells. Again, skin cells in turn are made up, can be of various types: adipocytes, epithelial, fibroblasts. Brain cells can be of multiple types, right? Astrocytes, neurons, epidermis. Right? Similarly, muscle cells. So, so in some sense, before single cell was discovered, which was I think 2009, we tended to kind of, aggregate all of skin cells as one kind of cell, right? And that has been the kind of power of single cell, that you can look at each individual cell, right? And different skin cells have very different structure and function, right? So just an example of pathologies and disease, right? So brain cells, neurons are implicated in autism and Alzheimer's. Skeletal muscle cells are implicated in muscle disorders like muscular dystrophy. Some of you might have heard of SMA, spinal muscular atrophy. Astrocytes are implicated in Rett syndrome, right? Fibroblasts are implicated in cardiac fibrosis. Fat cells, adipocytes, are implicated in obesity and hence by extension to metabolic diseases like diabetes, right? And dendritic cells are implicated in Crohn's disease. This is not an exhaustive list, right? So this is just right. So we'll kind of skip over this, but the two big wet lab platforms, I think the dominant one from what little I know of wet lab is a company called 10x Genomics. So you use 10x to actually do the sequencing and library prep. Basically skip over this. So this is an important slide, right? I realize not full screen. Let me go. So the top half, is on the x-axis is time, right? So 2009 is when the first single cell technology was invented, and I want I think the more interesting graph is the bottom one, right? So on the x-axis, on the bottom graph is still time. On the y-axis is number of single cells sequence, right? So if you look at this dot, right, the very first single cell study sequenced one cell, right? And that was 2009. And let's just zoom back to, so the graph only goes up till 2017. There's been way more progress since 2017. But now we are close to like 1 million cells in one experiment, right? And imagine what's been accomplished in the past 7 years, right? So it's just been astounding. Yeah. Okay. Okay. I assumed. Did you watch the video? Okay. 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 Good to know. We'll cover that. So I think we'll come to that. But at a very high level, we are saying that a single cell experiment in 2009 could sequence one cell. In 2017 could sequence an order of 1 million cells. And that's just like mind blowing, right? And maybe I think the number now may be more like 10 million or 100 million, right? So it allows us to probe perturbations in cell at a very high throughput and resolution. Right? So the dominant techniques, as in my limited knowledge of wet lab, is 10x genomics, and there is wet lab protocol. I think it's called drop-seq. Has anybody done any single cell? Wants to correct me? So the dominant thing used in industry and academia is genomics, and that's how you do the library prep. And coming to your question, right? So I'm going to use the whiteboard, right? So let's say you have a biological sample, right? Maybe a biopsy from a cancer tissue, right? Now you want to identify what, so I'm talking generally, not specifically RNA-seq, right? You want to identify what mutations are there, right, in that sample. So you take that sample, right, and you do some sort of, I would colloquially call it prep, right, which is preparing the library, which is the prerequisite steps you need to do to put it into a sequencer, right? And we'll briefly look at how a sequencer works in the context of single cell. Do you have a question? Right? So the dominant sequencing machine that is used is Illumina, right? And we'll not go into details, but we'll focus on Illumina sequencers, which do short reads, right, of the order of 200 to 300 bits. And there are now long read technologies called BIO and Oxford nanopore. So they can read much larger. So essentially what they're doing is, like, let's say, you take some sample of DNA or RNA, right? You cut it up into small chunks as part of the prep, right? Chunks or fragments. Right? Then you add what are called adapters. So, right, two colors. So, still two. So imagine this is one color, and so this is your fragment, right? DNA or RNA. You attach one kind of adapter to one end, to the 3' end, and one kind of adapter to the other end, 5' end, right? But I'll not go, that is the extent of my wet lab knowledge, right? And then you do PCR amplification. So, often you have like very few cells in the original biological sample you started off with. Then you'll do PCR amplify, and you will create multiple copies of it, right? And then is the interesting part of how, once you put the, so the sequencing machine is doing a bunch of these things, right? You are doing the cutting it up into fragments. Then it is doing PCR amplify, and then so I think for most of you who watch the video, it will be apparent. But imagine there are, like, from what I understand, a bunch of flow cells that are the kind of workhorse of the sequencer. And this thing is gonna kind of imagine like it's going vertically, right, along the y-axis, right? And at the end of each, imagine there's like it's hard to draw the 3D picture, but imagine that there's like a vertical, right, fragment of DNA or RNA, right? As it is passing through, right, the machine is trying to identify what base it is, right? In the case of DNA, A, T, C, or G. In the case of RNA, A, U, C, or G. And how does it do that? That as part of the wet lab and the chemistry of the sequencer, there'll be like a fluorescent dye, right? And there is some actually kind of interesting to me is that there is some computational aspect to it, as in, as the bases are going, right, there's an image being taken in real time, right? And you need 4 distinct colors to discriminate between 4 DNA bases or RNA bases, right? And as it is going, it takes an image and then does some image processing to identify which color that base is. And based on what color it is, it will identify it as a A, T, C, or G. But this process is not 100% accurate. So it can make errors. Over the years, it's gotten really, really good. So very low error rates, but not zero, right? And imagine like a thread, a thick thread, right, made up of beads. Each bead is a base, right? And in some sense, you are processing the thread bead by bead or base by base. Everybody following me so far? Right? So as each bead or base is being processed, you are recording it, right? And in the overall, there's a lot of software to reconstruct the sequence, right? Right? Like this was, let's say, the sequence of the biological sample. And the machine has an understanding of these flow cells, like where are they geographically located spatially located in the sequencer, right? So it knows that if there was a thick thread made of beads here and a thick thread made of beads here, how to join the sequence coming out of this and this and this. And if I'm not wrong, there are like 400 million of them in a high-end sequencer, right? And it can make mistakes, as I said, right? But there are sequencing technology has evolved so much that it's very good at correcting them. And it will we'll look at a small example of how the digital output of a sequencer, which is the sequence, right, which comes out of a sequencer, it's in a special format, right, made by an organization called NHGRI, National. standards are. So .bam, right? Or blanking on this one more, I think. You remember what's the other one? Yeah. So and if you watch the video, it would be apparent, but right, there are 4 lines for each small fragment. Right? So the first is some sort of identifier. Do people remember, was plus on the second line or the third line? Third line. What was on the second line? Right. So this is a unique identifier for that fragment. This is the actual sequence. Third line is just a plus. Nobody, I didn't dig into what's the significance of this plus. It's, I think, still obscure, like when they were originally, this file format was designed. And the fourth is some sort of quality score, which is a measure of how confident the sequencer was that this sequence is actually what was there. Right. So I'm going to pause here. A lot of, I think if you watch the video, most of it is a repetition of what I've said so far, but any questions, comments so far? Shireen, any questions so far?
Yes, sir.
Okay. Okay, let's, so coming back to the slide, right? Just to reiterate, exponential progress in terms of number of cells we can process. And the dominant technique, I think, is drop-seq, right? Droplet sequencing. So these are kind of the dominant protocols, right? So all of them have different trade-offs in terms of cost and throughput. So I think one of the rows I want you to pay attention is to cell capture efficiency. So I think the dominant is either 10x genomics or SQL, right? Or SmartSeq, right? We will, this is
What
just
does
like
this term actually mean?
Yeah, Shireen?
Cell capture efficiency. Like, what does it capture?
So I think may not be very visible to you, but imagine your biological sample made up of so many cells, right? Each of them is going to end up in a flow cell in the sequencer, right? So how efficiently were you able to capture, like record the number of cells in the sequencer using single cell RNA-seq?
Got it. Okay.
Okay. But I'll mostly gloss over the wet lab and protocol bits. We'll focus on the computational aspect. Okay. So bulk versus single cell, right? So these days the dominant is single cell, but we'll so in bulk, right? If you look at this figure, right? So each color represents a different sample, right? And it's like a collection or ensemble of RNA from so many samples, right? So, so when you measure or do further analysis on bulk RNA-seq, you get like, you don't get like, you know, the signal from the gene expression level from RNA sample green and the gene expression level from green RNA sample orange, and all the other colors are going to be like convoluted together, right? So you cannot delineate how much came from green and how much came from. So that's the power of single cell. But before single cell came around, I think most people used to use So bulk deals with averages, right? And for some applications, maybe averages are useful, but for most precision things, we use single cell, right? So, so if they came from these 4 different colors in single cell, came from 4 different populations or 4 different samples, you can delineate all 4 separately, right? So, so just to break down, I think we covered a lot of this already. So the first step is sample prep. Then you actually sequence, and then you do data processing and analysis. So data processing and analysis, I'll club together, right? That's the computational part, right? So I'm gonna skip this. So just one couple of terms you should remember is, for the adapter stuff, it uses something called UMI, unique molecular identifier, right? And it does, right? For each, if you look at this column of strings, right? So I think for different, so different genes are in different color, and there's a specific UMI which will kind of bond or aggregate to different genes, right? Or I think not different genes, to different samples, right? Skip this. Right. So now we are going to talk about what is the output of the sequence, right? Or basically the raw data, right? So imagine, so okay, basic question: how many genes do humans have? 20,000, right? So what you will get is right, a matrix, right? Gene 1, gene, right? And different tissues and organs and samples, like every gene may not be expressed in every sample or every cell, right? And that's what gives rise to the diversity of cells, right? Differential expression. So the rows of this matrix are the 20,000 genes, right? We're talking about humans. And the columns are samples, right? Sample 1, call it. And the very first video we watched said that normally for bulk, number of columns or samples is 6, usually 3 normal, 3 perturbed, or for bulk. For single cell, it could be anywhere, like it could be the number of columns. This is M, the number of columns in this matrix, right? Now, we have a matrix, right? So I'm going to use some terms. So this matrix will mostly be a what's called a sparse matrix, right? As in, a lot of the entries in this matrix might be zeros, right? Again, depends on the samples, but basically each entry in the matrix is read count per gene, right? What is the number of reads which map to gene 1 in sample 1? That is this entry, right? So let's say it is 10, right? So maybe gene 500 to 510 are all zeros. And 20,000 is, let's say, 500. So this is number of reads per gene, right? So you have got a matrix of 20,000 rows across, let's assume 800 samples instead of in case of single cell. So that becomes 16 followed by 5 zeros, 6 zeros back. So that's 16 million entries, right? So it's a fairly large matrix. Now that's not very, oh, sorry, yeah. Just press.
Hello?
Yeah.
Yeah, so we're saying that we have 500 of gene of gene 20,000
Yeah,
in one
hypothetical
sample.
but yes.
Okay.
Yeah, in sample 1, gene number 20,000, right? This will have a name. Let's call it whatever, RTG1 or whatever this gene represents, right? We are in sample 1, this gene is so enriched, so dominantly present in sample 1, that the number of reads you get for this gene is 500.
Okay, thank you.
This is just a made up example. I'm trying to motivate why we care about differential gene expression, right? Some of you who are already doing research, right? In our wet lab work, we always have some hypothesis, right? Like, for example, like TP53 is mutated in ALL, acute lymphoblastic leukemia, right? So what you would do, you would take a blood sample from a normal patient, you will take a blood sample from a patient with ALL, kind of blood cancer, and do single cell RNA-seq on both.
I can't hear sir properly. Then is the mic okay like earlier?
We can hear you fine. Maybe okay. Can you hear me now?
Yeah, better.
Okay. So I was saying, I'm trying to motivate why we care about differential gene expression. So in our research or wet lab work, we always have a hypothesis, right? Maybe gene TP53 is implicated in some kind of cancer, ALL, for example. So we want to kind of identify what is the genetic underpinning, right? This is genetic underpinning, right? And we'll do a differential comparison between a normal sample and a diseased or pathological sample, right? So we have everybody clear that we have output of single cell RNA-seq wet lab experiment is a file, which is essentially a huge matrix, which is 20,000 rows cross the number of samples in the, for example, in this case, 800 samples. So it'll have 16 million distinct numbers or entries of the matrix, right? So there are libraries in both Python and R to feed this matrix as an input, right?
Ashish,
Yeah.
these 800 are from 800 individuals, right?
800 samples, not necessarily. So the wet lab part is multiplex, right? So let's say all of us are working at Ashoka and doing single cell, right? You are working on cancer, I'm working on Alzheimer's, she's working on, I don't know, Parkinson's, somebody else is working on whatever, SMA, some muscular disease. And all of us are using single cell experiments for our normal and disease samples. So it's not cost efficient to run one run of the sequencer for you and one for me and one for her. So we will multiplex, right? We will prepare our samples separately, but they will go into the machine together, right? And the using barcodes and wet lab protocols, it can distinguish whether this came from sample, Anudar's sample or Ashish sample or Manasa's sample, right? Right. So it will, when you put it in the machine, it is multiplexing. When you get the data out, it is demultiplexing. So it can delineate that this number came from this sample. Okay. Pause here. Anybody have questions? So, so everybody clear that we have a matrix with 20,000 rows across some number of samples, number of columns, right? So 800 columns, which corresponds to the number of samples. So as I was saying, you can do it in R or Python. I would strongly encourage you to use Python. So in R, I think the library is called DE-seq. We'll focus on Python. So there's a Python library called scanpy, right? So we'll some of it in the second half, and in the remaining two classes, we'll actually take a paper focused on cancer and reconstruct figure 1 and some analysis using scanpy and Python. I'll share the data for single cell for that paper is publicly available, right? So you will download the data. So as I said, the community has done a great job of organizing, let's say, what's your name? Dia, right? Dia works on mitochondria. Can you maybe briefly talk about what kind of research you do? 1 line or 2 lines.
So my research looks at ovarian cancer, chemoresistance, and mitochondrial structure function in that context.
So imagine like there are research groups like this all over the world. So there have been people who have consolidated single cell data from multiple research groups into a corpus or a cell atlas or a single cell atlas, right? And they have like put data for, let's say, ovarian cancer and lung, like some other group focuses on lung cancer. So I'm going to quickly show a peek, and then we'll look at it in detail later. But I'm going to show you. So there's a fantastic resource called Cell Chroma, like X is cross. So CellX gene by the founder of Meta, Mark Zuckerberg, and his wife Priscilla Chan have something called CZI, Chan Zuckerberg Initiative, right? Okay, I am sorry, I didn't realize I'm not sharing my screen. Can everybody, maybe you can all open this also in the second half, right? So they have a catalog of 90 million unique cells organized across almost 1,500 data sets and 910 cell types, right? So there are various ways to explore it, right? So let's say I go to data sets, right? I can I'm interested in humans, and let's say I'm interested in, Alzheimer's, right? So it shows me all the data sets, right? So it will tell you what tissue the data set is, what disease it is implicated in, what assay, what organisms, and how many number of distinct cells. So if you just click on explore, so I've assumed you watched the video. So we are going to talk about how to do dimensionality reduction. I'm going to come to that, but I think since the disease came up, right? So now this is all the data, right? I can filter it by, right? So let's say disease. So there are multiple disease, right? So I have 11,000 cells for Alzheimer's disease, 11,464 for normal, right? And this tells me, right? So do you see the color code, right? The purple is diseased Alzheimer's cells and green is normal, right? And I can filter it to see if there are, sex-specific differences in Alzheimer's, right? So this is only male samples, and I can only look at female, right? I can filter it by, if you've read a little bit about Alzheimer's, right? The cells typically implicated in Alzheimer's are amyloid beta cells. So let's say if I want to, right? They have further 3 types, right? So I can look at, right? And we can try to find out if what disease. So these are, I think, ages. If I remember, like somebody who had Alzheimer's at age 57, right? 61. So imagine the sheer amount of, you could do all kinds of interesting analysis, right? So, and this is available publicly to everyone, right? So and this is one atlas. There are multiple atlas. There's an atlas dedicated to lung cancer. There's atlas dedicated to Crohn's disease. So if you're interested in generating hypothesis without doing any wet lab, just playing around with this data, you can get some understanding of what is an interesting hypothesis for me to explore, right? I'm gonna come back to the whiteboard now. Right. So where were we? We had a matrix with 20,000 rows, each representing one gene, and some number of columns, let's say 800, representing 800 samples. Right. Now, we have to, so the very first thing you can do is plot the data, right? This data is there, right? And let's say sample 5, right? So this is, let's say, normal, and this is sample 1 is ALM, right? Some kind of cancer. Now, having 20,000 genes makes things a little tough, right? To visualize, we are visual creatures, right? So visual things help, right? So hypothetically, if you had just 2 genes, right, you could immediately identify differences between gene 1. Now, if you have 20,000 genes, what you have is actually, you're dealing with a space with 20,000 dimensions, right? Instead of having humans can think in mostly 2 or maybe 3. Beyond 3, it's very hard for us to visualize, right? Even 3 is actually quite hard, I would say. So, so we have, there's a class of techniques called dimensionality reduction, right, which will take high dimensional data, in our case, 20,000 dimensional data, and compress it down without losing the inherent structure in the high dimensional space to a low dimensional space, usually with 2 dimensions. So within this class of dimensionality reduction techniques, we have linear. So the dominant technique used in linear, and which we'll focus mostly on, is PCA, principal component analysis. And then non-linear, the 2 main examples there are UMAP, right, and TSNE. SNE stands for stochastic neighborhood embedding. You don't need to know the acronym full forms, but imagine, so UMAP and TSNE are supposed to be slightly superior to PCA, but PCA is simpler, and mostly we'll focus on PCA. Yeah.
I have a doubt on like what are these dimensions? I missed that part. Like
so you are trying to so there are 20,000 genes, right? 20,000 rows. So they represent the 20,000 dimensions. You are trying to compare like gene 1 versus gene 2 versus gene 3 versus gene 20,000. So reasoning in 20,000 dimensions is very hard. Right? So what we'll do, so what happened, I'll give you a brief, I'll send you some more readings for PCA, or you can look up StatQuest PCA, which will explain it clearly. But basically, you are, it's a little hard to visualize, but I'll try. Imagine, so first think about 3 dimensions, right? You have X, Y, Z. In 2 dimensions, you have X, Y. So imagine a space where your data is in 20,000 dimensions, right? X, Y, Z, 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 like whatever, up till 20,000 different axes, right? Like this is 2 dimensions. You have 2 axes. In 3, you have X, Y, and Z. So imagine there was a space with 20,000 axes, 20,000 dimensions. I can't make sense of what is happening in 20,000 dimensions, right? So we are compressing the data in 20,000 dimensions to 2 dimensions, right? We'll call them PC1 and PC2, principal component 1 and principal component 2. However, the data behaved in the 20,000 dimensional space, it will still behave the same way, more or less, in this compressed dimensionality reduced space of PC1 and PC2. And there's a whole kind of computational way to go from a 20-dimensional space to a 2-dimensional space using an algorithm called PCA, principal component, right? There are 2 other ways. I said UMAP and TSNE. We'll focus on PCA in this class, right? Yeah.
Hello.
So very good question. What's your name? Sedu. So Sedu, excellent question. I kind of just waved my heads and said the behavior remains the same. So what it means is, in our context, which is differential gene expression, that so I need to explain something called clustering. So imagine, okay, I have, the whiteboard is 2 dimensions, right? But imagine I am drawing something in the 20,000 dimensional space, right? And I'm going to only talk about 5 cells or 3 cells, right? Imagine we had 1 million cells in our experiment. I'm going to focus on cells 1, 2, and 3, right? Let's call them 1, 2. So in the 20,000 dimensional space, don't try too hard, like imagine there were these 3 cells, and biologically, these 3 cells are similar. So in the 20,000 dimensional space, they are close to each other, right? So excellent question by Seru, that what does it mean when you compress them, but you don't lose the latent or inherent structure when you do dimensionality reduction? What I mean is, I do PCA, and magically now I am in 2 dimensions, PC1 on the x-axis and PC2 on the y-axis, right? So I'm gonna dim 1, dim 20,000, dim 2, whatever, 3 until. So now the power of PCA and dimensionality reduction is that cells 1, 2, and 3, right, also are similar in this reduced space. Even though the dimensionality has reduced from 20,000 to 2, right? The inherent or latent structure remains the same, which is very, like, otherwise this is almost impossible, right? How they will behave in 20,000, like I can't make sense out of this, right? Out of a 20,000 space. But if I'm suddenly given a 2-dimensional space, maybe cells 4, 5, and 6 clustered differently, right? And that is a hint to my biological hypothesis that 1, 2, 3 are different and 4, 5, 6 are different. They are different. The machine learning or data analysis jargon for this is clustering. 1, 2, and 3 clustered together, 4, 5, and 6 clustered together. So is there something inherently special or different which is causing 1, 2, 3 to cluster very differently and 4, 5, and 6 to? So so at a very high level analysis, maybe these are the normal cells and these are the disease cells, right? I had, right, maybe 1, 2, 3 came from this sample, the normal sample, and 4, 5, and 6 came from the disease sample. It's a lot to take in. I'm going to pause here just to catch some breath myself and pause for that. Use the mic.
So when you're saying we imagine it in the 20,000 dimension space, each dimension represents a different gene, and we're trying to understand how they relate to each other.
Right.
So then how would the samples come in?
So that's, I'm going to wave my hands and say that's the beauty of dimensionality reduction, right? That this, so in some sense, you can imagine that PC1 and PC2, PC1 is some linear combination of genes 1 to 20,000.
Right.
And PC2 is some linear combination of genes 1 to 20,000. And PC1 and PC2 are orthogonal. So in, I can't, here, in office hours, I can maybe go into, so into the nuances of how PCA works, but essentially we'll build something called vectors, and 2 orthogonal vectors are, in in two-dimensional space 90 degrees
Mhm.
apart, right? They are, the technical term is independent of each other.
Right.
Right. So, so imagine you have taken a linear combination of genes 1 through 20,000 and arrived at principal component 1. You have taken another linear combination of genes 1 through 20,000 and arrived at principal component 2. And you have built them in such a way that PC1 and PC2 are orthogonal, right? And since they are orthogonal, you can graph them in 2D space, right? And using data libraries like ScanPy and clustering, we'll come to clustering also, you can find interesting clustering patterns, right? Like 1, 2, and 3 are normal, 4, 5, and 6 cells are diseased, right?
Right. So the graphical representation would be like the diseased or mutated ones would be clustered around a
Clustered
certain region,
together, right.
and the healthy ones would be. Okay. Thank you.
But yeah, so that now as a biologist gives you either new hypothesis or proves or disproves the hypothesis you started with, right? Any other questions? Shireen, did you have a question?
No, sir, I'm fine.
Okay. Great.
It's still a little hand wavy to me how all of this happens.
Yeah, we'll, I think either post lunch today or in the next class, we'll look at details of PCA, and I'll share some readings on PCA also. Okay, so right, so I was talking about clustering, right? So there are multiple ways to cluster, something called k-means clustering, hierarchical clustering, and graph-based clustering. Can everybody see this slide now? So another thing is lineage tracing, right? So cells go through different states, right? So another thing single cell sequencing allows you to do is to, let's say there is state 1, there is an intermediate state, and there is state 3. And imagine this is like the time axis, right? So it starts in state 1, right? Then goes to this, then goes to this, right? And you see, right, all of them, like eventually as time passes, end up in either dark green or light green kind of cells, right? So that is another powerful application of single cell, that it allows you to trace the state transitions of cells, right? One, obviously, I said was differential gene expression, which allows you to check for differential expression between normal and disease states. So this is kind of binary, right? That gene 20,000 was highly expressed in disease, but maybe in the normal cell it was only 1 or 5, and here it's 500. So it is, we know that it is over expressed in disease. But you can also build what are called gene regulatory networks. So imagine like a graph. one regulates size, regulate gene size. These kind of complicated, we'll not go into details of this, but it allows you to do gene set analysis and gene regulatory networks. And like after this class, I'm gonna share a Jupyter notebook with code using the Python library called ScanPy for you to get familiar. Right? So so coming to, we'll not go into detail of this. I'll share a video and maybe talk about it in next class. But how exactly does PCA work? So we had this 20,000 cross whatever 800 matrix, right? So now this matrix can be composed, can be decomposed. So this was 20,000. This can be this process called singular value decomposition. So it can be so as a matrix product of 3 matrices, and the like I'm forgetting the terminology. There's like proper linear algebra terminology. So let's call them 1, 2, and 3. So 1 and 3 are orthogonal, right? And these help us arrive at PC1 and PC2. So when you learn linear algebra, you will learn to do this by hand. But in our class, it is one line of code, which is the line at the bottom, which is sc.pp.pca. So A data is the matrix and data. Get what NCOM stands for? I think number of So we'll do it using code. I don't expect you to understand the math behind, but it's like very formal math, which that if you look into how this function works in ScanPy, it tells you how to decompose. So now we'll look at a few examples, right? So for the biology people, there are PBMCs, peripheral blood mononuclear cells, right? So you do single cell, right? And when you do clustering, the clustering also assigns a different color to each cluster so that visually it is apparent, right? So, so can anybody, so you can tell very clearly, right, that the orange cells, which are CD14 plus monocytes, are very different from the rest, right? Can anybody tell me what's the relationship between these blue and red? Blue is CD4 T cells, and red is CD8 T cells. So are they more similar than like the rest, right? Because they are spatially together in the PCA, right? X-axis is PC1, Y-axis is PC2, right? So you could do this for, so this is, I think, some kind of NK or another kind of immune cells, purple. So this is somebody looking at immune cells from PBMCs. So one drawback of PCA is that if there is non-linear structure, PCA will not find that non-linear structure, right? So you remember I told there are two non-linear dimensionality reduction techniques, UMAP and TSNE. So the reason they were invented was because PCA is not perfect, right? Right? If there is non-linear structure, it won't show up in PCA. UMAP, and the second one is a bit of a mouthful, but again, very easy to calculate. I think temporal stochastic neighborhood embedding, small t, capital SNE. I'm not sure about temporal, just double check. Techniques, non-linear dimensionality, which are superior to PCA, right? Again, you in Python, it is one line of code. So it's not very hard to use them if you are writing code. So I'm going to skip this. So just a visual explanation of why TSNE is superior. So the top plot is PCA, right? And in this particular data set, there is a lot of nonlinearity, right? So if you see, it's not, PCA doesn't do a great job, right? It's very kind of mushed together, right? There's no clear demarcation. But if you do TSNE, you see very sharp clustering, right? Sure.
So if we did one PCA and we think that the data is more spread or something, then if we do the other thing, will it become more?
Yeah. No, so depends on the data, right? What is the inherent relationship within the data, right? All other things being equal, I would say use TSNE, because you don't know ahead of time whether there's linearity or non-linearity. TSNE will do the linear part also well and will handle non-linearity much better than PCA. Right? So it's a newer technique and it's a superior technique. Right? The math is more involved, but if you're not calculating it by hand, you're writing one line of Python code, then you don't care, right? So the math for PCA is simpler than math for TSNE and UMAP. So I'm going to skip this. I'm going to share these slides. If you want to dive into TSNE details, you can find any number of videos and slides on it. You use something called a similarity matrix over pairs from your high dimensional space and eventually arrive at a similarity matrix in the two-dimensional space, right? Low dimensional. And you calculate something called KL divergence, right? And I think the goal of the TSNE algorithm is to minimize the KL divergence. But we'll be hand wavy about it because our job in this class is to learn, right? So TSNE is also not perfect, right? It has, right? So imagine I have only two. I'm gonna do 4 combinations. So So there's a black cluster, there's a red cluster, there's a black outside red inside cluster, red outside black inside cluster. And imagine there are so many clusters as there is on the slide. So when you look at the whole visualization, that is called global structure. Right? When you focus on a small section of that, it's called local. Right? This is just jargon from clustering or data science or machine learning. So the drawback of TSNE is that, and imagine this was in the 20,000 dimensions. Now, when you run TSNE and go from 20,000 to 2, right? TSNE preserves local similarity. Like if in this small region, right, if there was this black and red cluster were close to each other, so this is local structure, right? When you go from 20,000 to 2, this local structure will be preserved. TSNE does a good job at that. But global structure, which is something which was here and something which was here, they are far apart in 20,000 dimensions. They may not be far apart in 2 dimensions, right? An ideal algorithm will preserve both global structure and local structure. I've thrown a lot of stuff at you. Are you getting most of it? Are you getting none of it? Are you getting everything? Some of it, okay. Yeah, it is, I think the 20,000 dimensions bit is a little, takes a little getting used to. And like ideally, I should include some more math. We have only like 3 or maximum 4 classes for the entire bioinformatics. So we don't have that luxury. Yeah. Right. So right. This you mean? Okay. Something like this. So if I give a slightly different definition, but maybe this is more precise, that local is, so this is light, imagine this is the light green cluster, right? So it says it's not very clear, but point within clusters are similar. So all the points within the green cluster are locally similar, right? So that will be preserved by TSNE, right? But whether these, right, let's say these which are far apart, right, and this, are they more dissimilar than this? We can't say when we use TSNE. Right? So now we have about half an hour before lunch break. So we're gonna quickly look at UMAP, and then I'll maybe share some code. So points which are closer to each other are similar, or cell. Each point is a cell, right? Each dot so this green cluster is made up of so many points, right? Each small dot is one cell, right? Similarity means that points that are closer to each other in this 2-dimensional space, either PCA or TSNE, are similar biologically also, right? Either, so you would, like if I were to look at this plot, I would say this is a distinct cell type, light green. This blue is a distinct cell type. This yellow is a distinct cell type. And I will say that the blue and yellow are quite similar to each other. I'll say these purple are distinct cell type, and they are very different from this and this. So this is tsne1 and tsne2. So as I said, you have done, you had 20,000 dimensions. That matrix was 20,000 rows. You have run some dimensionality reduction algorithm, either PCA or TSNE, or what we'll see next, UMAP, and now arrive at 2 dimensions, in this case TSNE1 and TSNE2. 2 dimensions we can reason about, right? For humans, we can comprehend 2 dimensions, right? So whatever, and I said, what is the key thing, key idea behind dimensionality reduction? That whatever was the global and local structure, or whatever was the relationship between all the data in the 20,000 dimensions, more or less that is preserved when you go from 20,000 to 2, right? And these 2, in the case of PCA, are linear combinations. In the case of TSNE and UMAP, are non-linear orthogonal dimensions or axes, right? Orthogonal means in 2 dimensions they are 90 degrees. This is TSNE1, this is TSNE2. So they are orthogonal to each other. And then we use some sort of clustering to identify this cluster is distinct, this cluster is distinct, and the clustering algorithm will, when you plot this, will also use different colors. So that it visually, like if they were all black, you will still realize this looks different from this looks different. But when you use different colors, it becomes apparent that they are different cell types, or they could be, like this could be high TP53 expression, this could be low TP53 expression, right? But you have some visual indication that this looks very different from this, right? Then you can trace back and go to your samples and find, was there, like look at, let's say pick 5 cells from this and 5 cells from this, and look at your values in the matrix, right? And see what, is there something dramatically, like, you know, maybe the 5 cells in this cluster, right? They all came from the normal column. These all came from the diseased column. Okay? Yeah. Right. Right. So, you know, if you look at, you know, this, the edge of the blue cluster and the edge of the yellow cluster, should this blue be actually be yellow, or could this yellow actually be blue? So at the cluster boundaries, there'll always be, they can fall either or that, but it will eventually come down to, you know, what clustering algorithm you use and what the data actually says, right? So what I'm not able to perhaps communicate very effectively is that there is some biological basis for what goes into blue and what goes into yellow, right? And even though they are not perfect, they do capture most of that, right? That blue is distinct from yellow, right? And even if something is technically, like if you look at these dots, right, you would imagine, like, we are trained to think that there should be clear boundaries. If you are different and I am different, like, we'll be neatly separated. But biology is not neat, right? That's the underlying message. Maybe even though this technically falls in the yellow cluster, for some reason, the algorithm classifies them as blue. So finally, we'll look at UMAP, right? So the acronym is Uniform Manifold Approximation and Projection. So I would say most cutting edge research, when you read a research paper, uses UMAP, right? Because it overcomes the limitation of TSNE also. It can capture nonlinear structure, and it is able to capture local similarity and global similarity. Okay. So here is like TSNE on the right, and UMAP on the left. Right? I think if I'm not wrong, this is not a biological data set. MNIST is the US Postal Service had a competition on classifying handwritten digits, but I'm not sure. MNIST, I'm going to check. I'm going to skip all the math, right? Right? So this is how you use UMAP from ScanPy, right? We are going to look at that. And finally, we are going to talk about the different, what's your name? Sedu. Sedu had an interesting question. How, whether it goes into the blue cluster or the yellow cluster, right? So there is some dependence on the particular kind. Like if you and I were analyzing the same data and you used k-means clustering and I used graph-based clustering, we might arrive at different visualizations, right? So there's some dependence on the kind of clustering, right? Usually people will adapt it to the particular kind of use case, right? So so these are the different kinds. So I think this is talking about graph-based clustering, right? So here you will kind of imagine it as a graph, and each, so okay, people may not, so a graph is composed of nodes and edges. Right? Edges represent the relationship between nodes. So this is node, and these are called edges. These are called nodes. Right? So when you apply graph-based clustering to single-cell RNA-seq, each node is a cell, and the edges between nodes are a measure of similarity, right? How similar is this cell to this cell is what is the edge in the graph, right? So there are other kinds of clustering, but I think the dominant one is graph-based clustering. I think these are examples of so there's something called KNN, K neighborhood, K nearest neighbor, right? And this is Lovain and Kuba.
So are we gonna learn how to cluster ourselves? Like
Yes.
those clusters?
Right. So, you know, so to take a biology example, right? Let's say you have French speakers and Dutch speakers, right? French is in red and Dutch is in green. So this is how, this is an example taken from, I think, phone calls on a Belgian mobile phone network, and they speak both French and Dutch, right? So you see that the French clustered together and the Dutch clustered together, and maybe this kind of middle part, which is zoomed in here, uses predominantly red, predominantly French, but some Dutch, some bits of green in there. So we are gonna, yeah, Shireen, to answer your question, we will look at code for clustering. Yeah. So and as you can see, right, in UMAP, I think it will automatically also assign distinct colors to each cluster, right? So that visually you can make out the distinct clusters easily. So I guess we are a little bit ahead of time today. Started at 10:45. So just to recap, we talked about how single cell RNA-seq works. We talked about how the data for a typical output, the file, the digital data output of a sequencer looks like. And then we talked about how it's very high dimensional. It's 20,000 genes. We can't make sense of a 20,000 dimensional matrix. So we do dimensionality reduction using two kinds of techniques. Linear technique is PCA, principal analysis. Non-linear are TSNE and UMAP. UMAP overcomes the limitations of TSNE. We are going to look at code samples, and you will fill up a Jupyter notebook with analyzing single cell RNA-seq data from a paper. So the disease for that data set is cancer. And you will try to reproduce figure 1 using that data set. So I'm going to talk more about what we are going to do after in the post-lunch session. In the originally, there was only 3 bioinformatics classes. I think we might do 4 because 3 would be rushing it a little bit. But I'm going to, after today's class, I'm going to share the paper. You have to, I'm going to share also a document called How to Read a Research Paper. So read that first. That is just 1 page, right? And then I want you to read it according to that document. There'll be multiple, you will do like a skim, pass 1, and you will do a detailed reading, pass 2 and 3. And then I'll share a partial with some of the code. And then I want you to kind of fill in the rest of the Jupyter notebook. And as part of the Jupyter notebook, you will download the single cell data from a central atlas using Linux commands and use ScanPy to analyze that data, right, in Python. So I think I'm going to talk more about the paper and stuff in the post-lunch session. And I think I gave you a warning. There'll be a lot of new stuff in lectures 2 and 3. So hopefully it was not all overhead, like you got most of it. And just like any difficult concept, you will, as you read more and as you digest it again and again, it will get easier. So yeah, there's no silly question. Any questions, doubts, comments? Yeah, I'll share this slide. And so there'll be a third class on 14th, next Saturday, and maybe a fourth class, right? The way the course is structured, that will be an optional class if there's one on 21st. So I can't require you to come in, but I hope if you're interested, you come. No. Modeling will be also 3, minimum 3, if not more. But we had, me and Sudipta had originally designed it as 6 Saturdays, 3 bioinfo, 3 modeling. But we might do 4 plus 3, with the 4th being optional, 4th bioinfo being optional. Yeah. So that remains to be seen whether we'll do the 4th bioinfo on 21st or the first modeling on 21st. Okay. I won't be there. I think 21st, to answer your question, 21st will be modeling. We'll figure out an alternative date for the 4th bioinfo. Right? It could be like a longer office hours instead of a class, right? I'll be on campus. I don't come to campus every day, so I'll make sure I'm on campus that day, and you can come with your code wherever you are stuck, or if something's not apparent, then we can either do like a mini class informally, or one-on-one you can ask your doubts. Well, right. So just that is more of a just to satisfy some Ashoka rule, because it cannot exceed a certain number of hours. So that is more like a formality than a. Because your normal courses are like sometimes twice a week or thrice a week for like an hour. And this is 4 hours in one day. So 4 times 6 is 24. So there was some limit that if you are going beyond 25, it has to be optional and not. But like there is so much new stuff being thrown, and some of you have not written Python code. So instead of being too fast, I'm just going to add the 4th optional thing, so that there's not, otherwise, whatever I was going to cover in 3rd and 4th, I'll have to cover rush through in 3rd. Did how many of you watched the playlist that I mentioned in my email? Right. Did you guys watch it? Yeah. There were 5. 5 together will take you 25 minutes. If you watch at 1x, you can watch it faster also. But I think those are, so the guy whose channel it is really good, Sanbomix, right? Sanbon. So he's like a genomics hardcore expert. So highly recommend it. And we'll be using the final video in that playlist is around Anaconda, right? So imagine like in Python, you have several modules and libraries. The commonly used libraries, they have clubbed together, right? this is just Python. This is Python, right? Now it has some standard libraries which are included. But let's say you want to install, I don't know, Excel or library to read Excel and PDF files called PyPDF. So all these you have to install separately, right? And for machine learning and numerical stuff, there's something called NumPy and SciPy. So if you want to use this, you have to install these Python libraries using pip install. So what Anaconda does is that the commonly used ones, it bundles them together. So Python plus NumPy plus SciPy, right? Scientific Python, numerical Python, and it calls it either Miniconda or Anaconda. So if you install this, right, you don't need to install each library separately, right? So we will actually use Miniconda or Anaconda. If there are no questions, you can break early and we can start early. We can start at 3:15, sorry, 1:15 instead of 1:30. Up to you guys. And end at 3:15 instead of 3:30. Any questions so far? No? Okay. So what time do you guys want to come back? 1:15 is fine?